Okay, I believe we're live, so let's start the webinar. A very warm welcome to our APMEN vector control community. Thank you for finding time to link in and share in these webinars. This APMEN group, vector control group, is a very special group of people. It's a genuine pleasure to interact with you all. I'm going to hand over to our APMEN Vector Control Working Group Chairman, Dr. Mohammed Mukhtar, to also welcome all of you. As most of you know, Dr. Mukhtar is head of the Directorate of Malaria Control in Pakistan. Thank you for opening this webinar, Dr. Mukhtar. Over to you.
Thank you, Dr. Liu. Very good day to you, respected vector control colleagues from Asia Pacific across the world. Welcome to our APMEN Vector Control Tech Talk, which is going to be on the technologies advances in vector control. As a reminder, this series of the ongoing Tech Talks is made possible by the APMEN, the Asia Pacific Malaria Elimination Network. As you know, maybe you know that APMEN has 3 working groups to support all 22 member states of the APMEN to achieve their goal of malaria elimination. This webinar today being our own vector control vector working group. Then there are this vector surveillance and the response working group, and finally the VIVEX working group. During the last year, we have now also established 3 cross-cutting interested groups. One is the climate and environmental impact on the malaria, and other is the cross-border issues and outdoor biting. We are in will be will be sharing our more detail about our next upcoming APMEN Vector Control Working Group annual conference, which will be held virtually this year during the first week of December. And we will the flyer announcing the conference will be shared with all of you very shortly. For today's webinar, why this webinar on technologies advances in vector control? Yes, you all know that vector control supported by the effective vector surveillance is the critical element in the combat against the mosquito-borne diseases like malaria and dengue. We face significant constraints in applying surveillance and control for various reasons, which include challenging finding the breeding sites, addressing the outdoor biting, capacity in the identification of mosquitoes, and practical surveillance tools and control tools that go beyond the limitation of the ITNs and IRS. We are going to have a no less than 5 presentations, 4 presentations today from the international expert, well-known expert across the globe to give us some insight on the advances in vector surveillance and control from the various perspectives. I hope you all will enjoy this very important webinar, and I'm sure you will have this. We will also will get some feedback in form of questions and feedback from all of you. Thank you very much, all of you, for joining. And now I hand over back to the Dr. Liu. Over to Dr. Liu.
Dr. Mukhtar, thank you for that welcoming message. And we understand your situation. You've just returned, I think just not many hours ago, you got back from a very long flight after attending the ASTMH conference in America. So very jet lagged, no doubt. So we wish you well and genuinely thank you for still being willing to participate in this webinar. Thank you for that. So just some comments on the webinar process. As mentioned, we have 5 expert presentations and we extend our sincere gratitude to each of the presenters. The 5 presentations will be given back to back and we'll then have a joint Q&A session at the end. I'd like to invite our audience members to please submit questions, but could I ask you that you please insert your questions in the Q&A box. If you look at the bottom, there's a Q&A box and there's a chat box. Please do not put your questions in the chat box, because there are a couple of hundred of you, and if you start putting comments and introductions and questions all in the chat box, it gets very confusing, and it takes time then to sift through everything. So please put your questions in the Q&A box, and I suggest you precede your question with the name of the panel member you would like to respond to your question. For example, Dr. Tony, and then your question. Then we know exactly what the kind of response is that you're interested in. After the final Q&A session, there will be a very short poll. It's quick, 1 minute of your time, but it helps us, and I'd be grateful if you can just quickly tick the various multiple choice responses to each of I think it's 3 questions. So it goes very quick. Please note that this webinar is being recorded and it will be made available on the Aurene website within a day or so. And the details of the website will be made available on your screens. Okay, let's get a move on. I don't want to bore you with any further explanations. Our first presentation is going to be by Professor Peter Hadawi, who will be informing us about integrating street view and drone images for detection of Aedes breeding sites. Professor Hadawi is the Global Talent Professor of Computer Science in the Faculty of ICT at Mahidol University here in Bangkok. He's also Honorary Professor of Medical Informatics at the University of Bremen in Germany, and is Director of the Mahidol Bremen Medical Informatics Research Unit. He has been a Fulbright Fellow, Hanse Wissenschaft College Fellow, and Avery Brundage Scholar. His research falls broadly in the areas of artificial intelligence and medical informatics, and he has published over 140 refereed papers, widely cited. For the last several years in a row, he was ranked the top computer scientist in Thailand and among the top 2% of scientists globally. His current work focuses on intelligent medical training systems and applications of artificial intelligence in vector-borne diseases. So we look forward to your presentation. Peter, over to you.
Okay, let me go ahead and share my screen. Okay, does that look all right?
Yeah, that looks perfect.
Wonderful.
And your voice is clear. Please go ahead.
Thank you, Leo, for that very kind introduction. As Leo mentioned, one of the things my lab focuses on is IT solutions to issues in mosquito vector-borne disease. And we look at vector monitoring, vector control, predictive modeling, and also diagnostic decision support. Today, I'm going to talk about one particular approach to vector monitoring, which is the exploitation of geotagged images to detect Aedes breeding sites. And I'm actually not going to be talking about the drone work, just because this is quite a short presentation. So I'll focus mostly just on the use of street view images. Although I'd be happy to answer questions about the drone work if anybody has them. So I think, as you all know, Aedes mosquitoes breed in small artificial water containers. And detecting and mapping such breeding sites is really essential to effectively targeting control, either by using larvicides or destroying the containers, or possible other measures. And of course, because of the importance, public health agencies do conduct container surveys, larva surveys, but these are done manually. And the problem is that because of the manual nature of this work, they don't scale well. So one does not typically, is not getting the data on a geographic scale that may be desirable, and also on a temporal frequency that may be desirable. So our objective and approach is to create highly detailed maps of Aedes breeding sites at the scale of cities like Bangkok and at the scale of provinces. And our approach to this is to automatically detect breeding sites in high-resolution geotagged images. Now, we have been working with Google Street Images, Street View images for this for a couple of reasons. They are readily available, they provide very wide coverage, and they're not always completely up to date. They're often a year or two out of date, which actually, if you want to pilot an approach, is not so bad, because then you can effectively align them with historical dengue case data and whatever container counts or larva counts we might have. But I'm also happy to address this issue of sort of the lifetime of validity of container counts. So how do we do this? Well, we take a map, electronically represent a digital map coming from a source like OpenStreetMap, and we have an algorithm that follows the roads and lays down pins, and we found every 50 meters works well. And then we basically pan in a circle and we pull in 6 images around for every pinpoint. And then we take all of these images and we run them through an object detection algorithm. When we started doing this work, we used something called faster RCNN. More recently, YOLO has become available and outperforms faster RCNN, so we use YOLO for this. The idea is the same in both of these models, which is that they will detect and classify different kinds of objects in the images. And so in applying this here in Thailand, and we have applied it in a couple of different countries. So in Thailand, we had to do some background research and find out what are the most important types of containers. And this is the list. So things like these large jars, ceramic jars, buckets, old tires, potted plants, bins, ceramic bowls, and then a couple of categories for miscellaneous open containers. This shows you the detection. So you can see here some potted plants, some tires detected, buckets, bins, etc. So this is the kind of data we get out of the detection. So then, of course, we need to evaluate how well this works. And there are a couple of different ways we look at this. One is simply to look at what is the accuracy of the detection algorithm. And here, what you have to have is some testing data that has been hand-labeled, where we can then see what the accuracy of detection is. And you can see that it varies a bit over container types, but the average, looking at the right, is quite high in terms of precision and recall. So we're quite good there in terms of detecting containers. And then, the question is, how useful is this now? Okay, so if we can accurately detect these containers, how useful is it to do this detection? Here is one evaluation we did. We did the whole province of Nakhon Si Thammarat. That's our first study site. And what we looked at, and we had some counts from manual surveys, not a lot, but we had some for a few sub-districts, and we compared our counts to the manual counts. And you can see that they correlate quite nicely, except actually for one sub-district, Khao Gao, and the reason for that is likely that the Street View image coverage was extremely low. It only covered a little over 1% of the area. And of course, this is an issue with using Street View, that the coverage is not uniformly good, But the good news there is that we know exactly what the coverage is. We know what the coverage is. We also know what date every image was taken because that's in the metadata that's with the image when you download it. And then we looked at comparing with larval survey data. And this is multivariate linear regression where we took the 8 container types as the independent variables, and the dependent variable was Berthaud index. And we looked at over the whole year, and the R-squared is about 0.3, which is not so great. But it also makes sense that it's not going to be so good because that includes the dry season when all of these containers contain no water. So if we look during the dengue season, the rainy season, now the R-squared goes up to about 0.67, which is actually quite good. Second study site, Bang district in Ratchaburi province. Here we happen to have some quite good data to compare to. This is just showing the number of images retrieved in every sub-district. This is showing now what we looked at. We had some very good data from the Ministry of Public Health on dengue counts, and we looked at the correlation between dengue counts and our container counts. And this is just taking all containers together. So what you can see is this is 2022 on the left, 2023 on the right. Our Pearson and Spearman values are quite good, 7.681, and on the right, 0.9, 0.88. So quite a nice correlation with the dengue case counts there. Another thing we wanted to look at was the added value of container counts in doing dengue risk mapping. So if we build risk maps, is it useful to include the container counts in the models? So we looked at Bangkok and Nakhon Si Thammarat during the dengue season. We had the dengue incidence, rainfall, temperature, population, and the densities of our 8 outdoor container types. And we looked at this at the sub-district level, looking at 3 models, 2 baselines, a generalized linear model, linear mixed model, and the linear mixed model plus the container counts as our experimental model. And what you can see here is the linear mixed model does better than the generalized linear model in 3 provinces. We actually did 3 provinces. And if we add the container counts to the mixed model, we do a bit better in every case. So what that shows is that there is added value to having this data. And the nice thing is that the data is essentially free, right? There's no manual labor. It's all just CPU time. So what are we doing now? We are working, as Leo mentioned, comparing the container counts we get from Street View to container counts we get from drone images. We're doing that in Rio de Janeiro with some colleagues there at the University of Rio who have been using drones to gather this data and looking at the container counts and also the complementarity of the data. We are also working on incorporating container counts into mechanistic models. So there are some models that use container counts, for example, Skeeter Buster. And those were used by manually getting container counts or by estimating container counts. And so we're interested in whether using the Street View container counts can be effective in these models. And we are also developing a fully automated software suite to better automate the process that I just went through because we haven't fully, so it's basically a single click and then everything just runs. And then we have one more technique that I didn't talk about today in terms of vector monitoring, and that is the development of bioacoustic sensors for adult mosquito counts, right? And so what we do there is we develop models that we can compress and deploy on small microcontrollers, so IoT devices, to detect and classify and count mosquitoes based on wing beat sounds. So that's, and just leave you with a little bit of further reading. We've been publishing this on a fairly good ongoing basis. So thank you all very much.
Peter, thank you so much. That was a fascinating approach that you demonstrated there, explained. I was very interested in the high levels of correlation between, your container counts and being able to relate that to dengue case numbers. I found that quite surprising, especially since, obviously Aedes breeds in very small containers, rain-fed bottles, tins, coconut husks, and so on. But it does seem as though your techniques, your approaches do recognize and count the more productive breeding containers,
Right.
which
Right.
are the bigger kind of containers, not necessarily the very small volumes of water.
Yep.
So lots of questions, I'm sure, that people would be interested to ask you. Great presentation. Thank you for that, Peter. We're going to move on to Christopher Nolte. And Christopher, his topic will be robust mosquito species identification from diverse body and wing images using deep learning. Christopher is a PhD student at the Bernhard Nocht Institute for Tropical Medicine in Hamburg, Germany. His research focuses on applying machine learning to analyze mosquito morphology, aiming to enhance vector surveillance. With a background in biochemistry from the University of Würzburg and health sciences from University of Applied Sciences in Hamburg, Christopher has a strong interest in vector research and image-based biological data analysis. So, Christopher, thank you to you as well for your willingness to share your insights, experience, knowledge. We look forward to hearing from you. Over to you, Christopher.
Wonderful. Thank you for the nice introduction. And yeah, I'm going to go a bit about the topic of mosquito species identification and how we can use artificial intelligence to support all of this. And usually at this point, I go through some explanation why it's so significant that we identify our mosquito species and that we can help our vector surveillance and vector control efforts with this. But I think in this webinar here, you may be familiar with all of this. And unfortunately, what motivates us is that the current methods we have to identify mosquito species are not up to our expectations. So usually either we use morphological means, which often requires someone looking at the mosquito under a microscope. And unfortunately, people trained in this are even more rare to find. And also, this is a time-demanding process. On the other hand, we have molecular methods, and while those molecular methods sometimes prove promising, they are not really up to our standards in terms of accuracy and of scope. And this motivated us to maybe think about another method for mosquito species identification. And we are a research group at the Bernhard Nocht Institute in Hamburg, and we look at disease modeling, but also we are really interested in wing morphometrics. And maybe if you're not familiar with the topic, is that in wing morphometrics, we can use landmarks and some key features on the wings of mosquitoes, especially the veins, to separate or to investigate mosquitoes. So with a few landmarks set on a wing, it has been found in research over the last 10 or 20 years that we can not only find interspecies variations, so for species identification, but that we even can separate different populations using this means. And I'm researching to automate such methods and to use artificial intelligence to maybe utilize those key features in those wings and those morphological features in those wings to support our efforts of vector surveillance and monitoring. And similar to the previous speaker, we are relying on convolutional networks or on computer vision methods. And usually, I will maybe not go into the detail of the model, how it works, but generally we're using a model which takes an image as an input, then transforms this image, or learns to transform this image into only a representation of its useful feature, what we call a feature map. And then this feature map is then further analyzed by a dense neural network, which then can give us a prediction on the species, which was our first goal to see how much information we can get out of those wings. And the first question we asked ourselves is, how can we best do an image of a mosquito so that an artificial intelligence is able to analyze this? And we did this in a small scale experiment where we took 4 different mosquito species and image them with different devices. So we utilized just smartphones, then smartphones with just a small attached macro lens, and also then a classical stereo microscope. And we did this for the body of the mosquito and the wing of the mosquito. And we generally expect that the wing of the mosquito may work better for the classification of using the artificial intelligence, because we expect less variance in the images. The mosquito can have maybe some parts missing, maybe the color is changed, or the appearance is changed depending on the age, and you could put the mosquito in very different positions. And those are all positions the artificial intelligence, the convolutional neural network, has to learn and has to adapt to. And in the case of the wing, it's a nearly 2-dimensional object. You can only place it in one certain way and image it. And therefore, we expected that the wings may work better, even if they require a bit more work to get to them. And during our experience, what we've seen is that those generally that the convolutional neural networks use are a good method to identify mosquito species. We reached across the board an accuracy over 80%. And furthermore, we also see we see results that align with what we expected. So wing images generally show improved performance up to 90% accuracy on a relatively small training set of around 2,000 images. And also they require less images to be trained on, which is also really nice. Also really interesting for the further application is that even the images done with a smartphone result in good performance of the model. So we don't need an expensive stereo microscope with a camera attached to do the images of the mosquito, but you can use just a regular small build with a smartphone, and you can image both the body, and if you have a small macro lens, which costs $5 to $10, then a petri dish, you can even image those wings using your smartphone, which is really promising for our application. And lastly, and more the most interesting thing we found is that our performance in the controlled setting does not really align with the performance in the real world. Those artificial intelligence networks are really, those CNNs are really good at classifying images which are familiar or are looking familiar to those images it has been trained on. But if I use, for example, another device or maybe other lighting conditions, the model starts to fail quickly and starts to give us lower performance than we hoped for. And we put all of this into a nice publication if you're interested in where we compared the performance, and which we will go more in detail on the results. But I wanted to show you how we've gone from this study to maybe now a usable application. Because motivated by this and motivated by these results, we collected a large data set of mosquito wing images. So currently our data set consists of around 18,000 images of different species, mostly of the European distribution. So if you want to change that, please contact me. We are really open to collaborate on all of this and to include all of this. And we in this last week, we published our data set, so everybody can access it and everybody can utilize it and maybe even do their own experiments on them. And but what the problem with this data set is that it's very heterogeneous. So it has a lot of different imaging devices. So here are just a like small part of our data set which you look at, and you see that those like imaging characteristics like deviate from the different project we've done in the past. And this is a problem because you have to imagine that the convolutional neural network is a bit lazy. It will try to find like patterns in those images to associate a wing with one species. But does not have really an understanding what it should look for. And it will just look for the easiest feature to extract. So for example, if I image just one species always on a blue background and another species always on a green background, the model will never do the effort to learn maybe the characteristics of the species, but it will just use the easiest feature, the background and the color. And because we don't want to like introduce or at least mitigate the impact the bias of our data set has on our model, we've come up with an imaging preprocessing pipeline which takes those swing images, removes the backgrounds, removes some kind of the angle, removes color information, and then we use some histogram equalization measures to even reduce more features from those images. And to prove how well our imaging processing pipeline reduces the impact of the bias of our dataset, we did a small experiment where we trained our models just on one device and then tested them on another device. And what we generally see in our results is that if we have unprocessed data, we get really nice performance on the device we've trained, but the performance drops off significantly when we are going to try them on a new device. But with like our increasing pre-processing and with the increasing removal of undesired features of our image, we can like reduce this gap to an acceptable amount, which is really nice and which really helps us to build an application which can be not only used by us on our microscope, but can be used by other people around the world. And then we used our imaging preprocessing pipeline, used our large data set and trained a model. And what we got is pretty remarkable results across the board. So we see a balanced accuracy and an F1 score of around 95%. And even more interesting is when our model is doing like misclassifications, often does it with morphologically very similar species. So it does reasonable mistakes. And that we see also in our top 2 balanced accuracy, which goes up to 98%. So you can be 98, at least from the testing data set, we see that in 98% of the cases, the model was either predicted with the highest probability or even the second highest probability. And we've wrapped up like our imaging preprocessing pipeline and our image classification model to a small application, which I hope we can make public in the next week. We're currently working with our IT department on them, but it's just a very simple application where you can just go through and just upload your images of mosquito wings. And if you upload them, the model will classify them, and you will get a list of predictions of the model, and which tells you which species it's most likely, and maybe which species it thinks it's second most likely. And you can even download then your data as a CSV file and maybe do some analysis on this. And to also see if this works like in a real world environment, we asked some people, we gave them mosquito wings and asked them to image them independently. And what we generally see is that the model is really good, even if the observer or the person behind the microscope is changing. So the model does mistakes, and we got an accuracy of around 92%. But when it does mistakes, it always does the mistakes on the same wings. So this gives us hope that our model is somewhat reliable in regard of the user. And this now maybe summarizes, or to summarize our work, is that I think we could underscore the potential of convolutional neural networks for the advancement of mosquito species identification. And if you're maybe not familiar with wings, this may be with the study on wings, it's maybe a bit like unfamiliar to you, but due to our previous work, we are really satisfied with our results and see that those wing images hold like really great promise, maybe compared to the body images of a mosquito, which may require less work. And we believe that we developed a robust imaging preprocessing pipeline which is able to reduce bias and create a system which is adaptable to multiple scenarios, so that it's not like just an in-house solution, but maybe a solution which a lot of other people also can use and profit from. And in the end, we present a classification system which is pretty easy to use, which does not really require expensive materials, and which only works based on a cheap macro lens, which you can attach to your smartphone with really, acceptable results for us. And most interestingly, we now have a model which is capable of, a system which is capable of analyzing wings of mosquitoes. And we are currently thinking about not stopping at mosquitoes, but maybe going to other vectors such as sandflies. And this wraps up my presentation, and thank you.
Well, thank you for sharing your insights and experience there, Christopher. I mean, it's absolutely critical to be able to identify which mosquitoes you're working with. There are more than, multiple thousands of different kinds of mosquitoes, and only a relatively small number of them are effective disease transmitters. And of those small number of effective disease transmitters, malaria, or dengue, Zika, chikungunya, etc., each of those has different behaviors. Some bite indoors, some prefer biting outdoors. Some rest indoors, some rest outdoors. Some breed in small pools, some prefer to live in streams. Some will readily bite cattle, some readily bite humans. So to know which mosquito you are dealing with in a particular setting, you have to be able to identify it so you can use the appropriate control approaches and understand why your particular control strategy is effective or not so effective. So thank you for those insights, Christopher. We're going to move on to our third presentation, and we're fortunate to have Professor Luc Alfie. Luc is a global leader in the emerging field of genetic pest management, focusing particularly on mosquitoes. He is a professor in genetics at the Department of Biology of the University of York. He co-founded Oxitec, which says a lot for Luc. Oxitec is a very highly regarded commercial company who's done wonderful things. We can talk about that in another talk. He co-founded Oxitec, a spin-out company from Oxford University in 2002. At Oxitec, Luc successfully led the world's first releases of genetically modified insects. Since moving back to the public sector in 2014, his research focuses primarily on enhanced sterile male methods, local gene drives, and anti-arbovirus refractory genes. So Luc will inform us about gene drive technology for mosquito population suppression or replacement. Thank you for being willing to share your insights with us. We hand over to you, Luc. Thank you.
Thanks, Leo. Let me just share the screen. Okay, so I guess conflict of interest thing. I no longer have any financial interest in Oxitec. I do advise two other small companies, Biocentis and Synvect. Okay, so I'm going to talk about genetic control in general and mostly on gene drive technology. So what do we mean by this? So I'm going to talk a little bit about what do we mean by genetic control, and then the potential outcomes of suppression or modification, and then on to gene drives. I'm going to talk a little bit about what my lab does, but only a few sentences, mostly about the field in general. Excuse me. So genetic control or genetic pest management is the whole feature is about releasing some modified version of the pest insect, which will then mate with the wild pest population and thereby deliver some heritable trait into that target population. This dependence on mating makes it extremely species specific, which is very attractive from an environmental perspective. But of course, if you have multiple vector species, as may be the case in some malaria settings, for example, then that's potentially an issue for Aedes aegypti, dengue, and so on. It's really Aedes aegypti is the key vector in most locations, so you do have a single species. But this will vary from one place to another. And it's a common feature of all these genetic controls, very species specific. Of course, you also have the modified vector going out and looking for the pest for you. So while it's very useful to know breeding sites and all that kind of thing, which will tell you about density and location and so on, actually the mosquitoes will go and find each other pretty efficiently by this method in a way that sprays and so on won't do. It's an area-wide control method, so the released mosquitoes will control the pest over the released area and potentially with gene drivers beyond that. And so this again has an effect on the economics. If you're talking about a very dispersed rural pest, you might need rather powerful genetic systems to get the coverage with modest release numbers. Urban pests, it's a little simpler from an economic perspective. Very good at reaching hard cryptic populations because, as I say, the mosquitoes, let's say the male mosquitoes, seek out the female mosquitoes rather efficiently. They don't have computer vision; they just have really good evolved vision and sensors. And it's equitable in the sense that everybody in the control area is equally protected, irrespective of wealth or other characteristics, which was one of the attractive features that got me into this area in the first place. So then what do you do with this? If you can introgress some heritable trait, what's it going to be? And although there are very many traits one can imagine, the ones in current use basically boil down to two different types of outcome. One is population suppression, where you're just trying to reduce the numbers of the vector population by introgressing a trait that affects biological fitness in some sense, so viability, fertility, or whatever of the population, which will then reduce their reproductive potential, and then by control them over time. And these are usually multi-generation approaches, but a generation is about a month for many of these vectors, so fairly rapid outcome, but not instantaneous. Population suppression is what most conventional vector control aims to achieve by different means, and it stops transmission. So if you think about Aedes aegypti transmitting a whole range of arboviruses, suppression would stop all of them. Modification is a bit less familiar. It aims to modify the target population in a way that prevents transmission. So this is the sort of equivalent outcome. What if you could vaccinate the mosquito population against malaria or against dengue or something like that? Of course, you can't for all sorts of reasons. But if you imagine what that would look like if you could, then you would still have that target population there, still filling its ecological niche, still biting people, but no longer transmitting the disease, or at least less able to transmit the disease. So it's a rather different sort of outcome, and it has pluses and minuses. And then, if you were a control program manager, and some of you are, or a political leader or whatever, which would you prefer? And there's no right answer. The different ones are better or worse in different circumstances. And part of the aim of developers like me is to produce options so that communities and program managers can choose the outcome they prefer. At the moment, there are rather limited options. Oops, sorry, I'm going the wrong way. Okay, so what's actually in the field? Talk about limited options. What's actually in the field at the moment? Well, there are a number of sterile male type methods based on historical use in agricultural pests with radiation sterilized males. Those seem not to work especially well with mosquitoes, but replacing radiation with genetic methods has allowed very successful use of this approach, of which the leaders are probably genetic modification methods from which I pioneered at Oxitec, but there's also PGSIT and a number of others, or Wolbachia-based sterility, IIT as it's sometimes called, used, for example, in Singapore in a very successful program there. So sterile males are extremely effective, but you need to release a lot of them, so you need to overwhelm the target population so that most wild females mate a sterile male, and they don't live very long, typically less than a week. So you need to replace that population on a very frequent basis. So much of the motivation for gene drive methods is to try to get somewhat similar effects, but requiring far fewer mosquitoes. And you know, my group is working on this both on gene drives, but also we have developed a much more efficient sterile male system, which in mathematical models, including other people's mathematical models, would require about 20 to 100 times fewer mosquitoes. And if I can have a little plug, we are looking for potential field trial partners. So if you know anybody who might be interested, please put them in touch. And then in terms of population modification, the only thing that is out there at the moment is the Wolbachia population replacement system, or Wolbachia gene drive system, which aims to both spread itself into the wild population and also reduce the ability of that population to transmit dengue. And multiple field trials have indicated that it can, at least under some circumstances, indeed persist and reduce vector competence, although there also have been a number of failures to persist. Okay, so if I just pick out a few words from that, the Wolbachia, not always called a gene drive system. So if I just pick out a few words from what I just wrote there, manipulate their host reproduction to allow them to spread through the population despite not conferring individual fitness advantage. And that is pretty much the definition of a gene drive system. So a gene drive system is a heritable system which can bias inheritance in its favor so that it can spread without necessarily providing individual fitness advantage, which is in sort of Darwinian evolution, what we think of as how genes, alleles increase in frequency. And there are many, many naturally occurring gene drive systems, transposable elements, Wolbachia, meiotic drive systems, a whole bunch of different ones. And synthetic biologists take their inspiration from natural gene drive systems to build synthetic ones on similar principles. The Wolbachia one is not a synthetic one; it's a transplanted one from the best known one from Drosophila into Aedes aegypti. Okay, so these are gene drives, things with bias inheritance in their favor, allow them to spread. And you can classify them in a number of ways. I talked about one is for all genetic control systems, how persistent are they, or how invasive are they, or how likely are they to spread? And you can think of this on a spectrum. On the left-hand side, things like sterile males, effective, but you need a lot to release a lot of them. They're very controllable, very reversible. So if you stop releasing sterile males, whether it's Wolbachia sterilized ones or genetically modified ones or whatever, they will disappear from the environment very quickly. And so they're controllable and reversible, but also rather expensive to deliver. At the right-hand side, you could imagine a highly invasive gene drive system, which from only a few individuals will spread from one population to another and by migration through the entire species. So although the development and regulatory cost might be high, the deployment cost would be very low, but it's also rather hard to control and reverse. And then in the middle, potentially you have what we call local gene drives, which might persist, which will persist in the target population, but not spread beyond that. And my group's working on these quite a lot, and we see them as a nice trade-off between controllable and reversible and lower cost deployment than sterile males, but more than invasive gene drives. They're also perhaps the hardest to develop. So what do I mean by local? So imagine you have a set of populations connected by a degree of migration, and you release your modified mosquitoes into one of them. So release into that target population, then what might happen if you stop releasing? Well, one possibility is that you just the mosquitoes disappear. So self-limiting systems, sterile males, for example, just will just disappear. Another one is if it's a highly invasive system, that small degree of migration from one population to another will lead to it being introduced successfully into other populations and spreading and invading in each of those. We might call these global gene drives or non-local gene drives. And the final population is that the gene drive will persist in the target population, but not spread substantially beyond that. And those would be local gene drives. And again, which would you prefer? And again, there's no right answer. So you might prefer to deal with the entire species at once, although a certain amount of agreement between governments might be required for that. Or you might prefer to target the particular population, and you might prefer something that you know is reversible. If you know if you stop, it'll go away very quickly, or that will persist, and you don't need to maintain it. There are advantages and disadvantages to each of these. Okay, so I've already said what's in the field, which into gene drive terms is basically Wolbachia, and then also sterile males at the more reversible, controllable local level. And then in lab development, there are enhanced sterile mail methods, but there are also gene drives. So probably the best known is target malaria for Anopheles gambiae, sub-Saharan Africa. And this is aiming to develop an invasive gene drive that would, in principle, spread through the entire species and possibly through the related cryptic species. I'm sure you know Anopheles gambiae sensillatu is a set of very closely related species. But there's a limited interbreeding between them, and that might be enough for a gene drive to spread from one to another. And then in terms of population modification, again, there are some gene drive approaches there. Again, the best known ones are the most advanced ones, all for Anopheles gambiae, and that's the University of California Malaria Initiative. And they're trying to spread a gene drive system that would lead to the mosquitoes expressing anti-plasmodium antibodies and being less able to transmit malaria. So a modification drive rather than a suppression drive. And then Transmission Zero, based at Imperial College, I have both local and global designs trying to spread antimicrobial peptides that would again prevent or reduce the ability of Plasmodium to infect those mosquitoes. And my group's developing local drives rather than those global drives for Anopheles stevensii and Aedes aegypti. And we do have a couple of PhD students, one advertised, one not. If you know anybody who might be interested, please put them in touch. And then the question that I always get for these, or therefore preempt, is will anybody actually use these? What's the public opinion about the use of GM mosquitoes? And I think this will vary considerably from one place to another. This is very early work. This is more than 10 years ago. So when releases of genetically modified mosquitoes were extremely new. And this was in our first project in Brazil. And you can see the name of the project. I have no Portuguese, but it's pretty clear what we were talking about there. And this is from a survey by Brazilian scientists in two areas. Itaberaba is a sort of high-density suburb, which is a poor suburb of a northern city, and Mandacaru is a village outside, so rather different demographics. And I'll just draw your attention to this one question. Do you want the project to continue with releases in this community? So this was after we've been going for a little while, and 97 and 98% of the survey people said yes, they did want releases to continue. So that doesn't mean that everybody everywhere will like genetically modified mosquito approaches, but clearly, at least in some places, you can get very, very high levels of approval for this kind of approach. This was sterile mails, a form of engineered sterile mail. And that's it. Thanks.
Well, you say that's it. That's more than just that's it. That's a very big mouthful. But it's a fascinating, absolutely fascinating topic, Luke, with so much potential and promise for very narrowly target species-specific interventions. And then, you know, kind of a forever after option built in where you release some and then it just spreads, whether locally or globally, according to your preference and the particular genes you've introduced. But it holds so much promise. So I'm sure a lot of people are going to ask a lot of questions. I certainly have a bunch of them, but you know, let's give the audience priority. We'll get to that. I'd like to remind our audience, because I'm sure we've got a whole bunch of new members that have joined. Not everybody joined on the hour. May I ask that if you have questions, please put your questions in the questions box, not the chat box, because if you put it in the chat box, it gets diluted with you know, people introducing themselves and making general comments and so on. And we're only monitoring the Q&A box for questions. So if you have a question, please put the name of the panelist that you would like to respond to your question, followed by your particular question. That makes it easier to direct your question to the appropriate member you'd like to respond to your question. Luke, I can just mention that I think it's in the chat box. There was a request from somebody for your, it seems the person is very keen to make contact with you, and he's asking for your email. So it's up to you whether you're willing to make your email, or you can respond directly, privately to that person. You don't have to make it public. So we move on to our next presentation, who will be by none other than our chairman of the APMEN Vector Control Working Group, Dr. Mohammed Mukhtar, who will be telling us about drone-assisted vector control in Pakistan. So it's just as well you didn't talk much about drone use, Peter, and you focused on the street view, because now we've got Dr. Mukhtar that can focus more on the drone technology. Dr. Mukhtar is the director for the Directorate of Malaria Control in Pakistan. He is responsible for overall management of the malaria control program, including to develop national policy, strategy, and guidelines for all vector-borne disease control. He's also the national focal person for vector-borne diseases, public health, pesticides management, integrated vector management, IVM, and for neglected tropical diseases, which embraces a whole bunch of different diseases, including helminths, well-transmitted helminths, and so on. Currently, he's a member of the WHO pre-qualification team for vector control products assessment, and also a core member of the Alliance for Malaria Prevention, AMP, in Geneva, and also the RBM Vector Control Working Group in Geneva. So, Dr. Mukhtar, we look forward to your experience in the use of drones in Pakistan. Over to you.
Thank you, Dr. Liu, for nice presentation. Can I share my slide?
Please do. Please do.
Yeah. Oh, it's not showing somewhere. Sorry, the slide is...
No, take your time. Take your time. No panic.
But why it's not showing the slides? Yeah.
Dr. Mukhtar, I don't think your slide is coming up. You want to try again? Stop share screen and try again. Yes, there we are. Okay, can you make it full screen, please, Dr. Mukhtar? There we go. Wonderful. Okay, over to you. You're on mute, Dr. Mukhtar. Dr. Mukhtar, you are on mute. Can you unmute yourself? You had it on screen. It was working at one point, Dr. Mukhtar. I think if you unmute yourself as well, then we can discuss your problem.
Is it okay now?
Yes, okay. At
Yeah.
least we can hear you now, but we don't have your presentation on screen. There we are. Okay, if you can make it full screen.
Yeah, it's okay now?
Perfect. Okay, please go ahead, Dr.
So
Mukhtar.
thank you very much, Dr. Liu, for a nice introduction. And today I will elaborate the Pakistan first field pilot project on use of drone for the surveillance and control of particularly the dengue in Pakistan. In the beginning, I will like to have a very wonderful thanks to our partner organization, UNICEF Pakistan country office, who supported this project and make it possible to have a Pakistan first pilot project on the use of the drone for the vector surveillance and control in Pakistan. Just a few words on the background. because my presentation are totally based on our field work, where we are facing some big issue of the control of the dengue in the country. You know that the use of drone for the vector surveillance control is an innovative and progressively more trendy method for the monitoring and control of the monitoring of the population and overall management of vector-borne disease in various scenarios. Drone give us a number of advantages in vector-borne disease management program due to their ability to cover a large-scale area in a shortest possible time and collect a real-time data and operate in an area where it may be difficult or hard to reach. This is the actual point for which we launched this project, because there are certain areas in the country, particularly the inaccessible breeding site for dengue control, and which is one of the major identified reason why dengue is always there is regular outbreaks, particularly in the capital, just because of there are certain places which are difficult or hard to reach or inaccessible to for the normal surveillance and control. So this is one of the main background of my this pilot project in the country. The main use of the and benefit of drone and vector control, it give us a vector surveillance of the vector population. Drones equipped with the various sensors, infrared, thermal, and multispectrum cameras that carried a high resolution map of the environment, identified the potential breeding site for the vector to in particular in the areas of difficult to reach. They can be combined with GIS, Geographic Information System, to analyze and mapping of the vector population dynamics and habitat of the time and space, which facilitate to develop a concrete strategy to control these to manage the breeding site and overall the management of the dengue vector-borne diseases, particularly dengue. And the other way, it give us a very precise application techniques in a very precise manner, minimizing the drift and reducing the environmental impact. And this method is more efficient, particularly in the hard to reach areas like in our scenario. Release the biological control, which my other colleagues also mentions, then monitoring of the efficacy of the program. It gives us a very good hand to monitor our ongoing activity. So these are the just a few backgrounds which build us give us the direction to build our pilot project in Pakistan. Now I come to my actual point. Pakistan, being a subtropical country, has a rich fauna of the mosquito fauna, of which many are the public health importance. Vector-borne diseases are of public health importance in the country. Currently, we have the major public vector-borne disease, the malaria, dengue, chikungunya, leishmania, and Congo. However, due to climate change and some other factors, which can be discussed in some other area, other topics, that the Japanese encephalitis, West Nile virus, and yellow fever are particularly upcoming threat in the country because of this climate issue in the country. Pakistan, in dengue, particularly dengue in Pakistan, is the fastest emerging arboviral infection, which I explain in this slide. You can see this is just the comparison or the background of the study, why the dengue is a so big issue in the country. There are two scenarios in 1994 to 2004, when we have only 4 districts out of 154, and the population, 6% population at the risk during their early 10 years. And total suspected cases during the 10 years from 1994 to 2004, only 3,000, and confirmed cases were only 700, and deaths during 10 years. However, next 15 years, dengue spread so quickly in the country that the 145 district, jumping from 4 district 145 just in 15 years, and 100% population at the risk. 3,000 suspected cases jumped to 18 million, and the confirmed 700 confirmed cases to 300,000, and the number of deaths increased to more than 2,000. This shows that dengue is the most rapidly spreading mosquito-borne arboviral infection in the country. The same is that the vector population. You can see the container index, and almost 3 times increase from 1994 to 2022. So Again, the dengue vector is also rapidly spread in the country. This is just the explanation why the dengue is so quickly spread in the country. And you can see that the container index during 2094 to 2004, and it from 7.5 to it jumped to 25. The same is other indexes. This shows that the dengue is a dengue vector is most rapidly spread in the country. Now, why we use this drone project in the country? One of the identified risk factor for this huge and regular outbreaks in particular in Islamabad is the dramatic increase in the illegal and unregularized business of garbage collection and storage in the periphery of the Islamabad. You can see the right side, the pictures. These are the empty bottles. It's a one site. And we have identified the 11 sites in the periphery of the Islamabad. Mostly this illegal business, because illegal business totally in the open places. There is no shelter to protect this business from the rainwater. And 341 families directly linked with this business, and 733 indirectly identified with this business, which means that this is a very big business, though it's illegal. No, it's not a regularized business. And those people who are collecting this garbage, they have 10 to 25, 23 water storage container at their own home, because they have a lot of plastic containers, and the which they found the container good, they keep it at their home for the storage of the water. And out of these, almost 90% without any coverage, while normally we have 2 to 3 container per household in our rural areas. But these people who are engaged with this business have 10 to 23 water container at their home. The identified business place have some local shredders, but they are not shredding the garbage over the months. So though they have some shredders, but they don't use these shredder, and they keep this garbage over months and months. And one very good example, that 330 to 35,000 mineral plastic bottles are produced one 4-star or 5-star hotel in Islamabad, and where those bottle goes. And some hotels have their shredders but not following the SOPs. And overall, the result shows that the 30 to 35 to 43% of these water containers, empty bottles, are positive with Aedes mosquito. Now you can imagine how much high magnitude is the positivity rate in these empty bottles. And these are some pictures. You can see these empty bottles with have some waters, and these are all are positive with the Aedes mosquitoes. And why would you? And just I give you the is want to show this one video, which you can imagine the magnitude of this business in the periphery of Islamabad. Now you can see the huge garbage piles around the capital. And just is a 30-second video, and then I will continue our use of the drone, how we use the drone for this surveillance. Now you can see these pictures. And all bottles are lying without any shelter, and they are placed in the open. And when there is rain, these bottles are filled of water. And the water is filled, then Aedes mosquitoes start breathing. So you can imagine the size of this business in capital. Now, why we use this drone project? National Malaria Control Program, National Entomological Reference Laboratory Network developed a proposal for pilot project for use of drone, particularly in these inaccessible areas, because the normally people don't allow you to go these areas because this is illegal business. So they hide this business from the local authorities. That's why first we make a drone surveillance to identify the areas without having the very direct in contact with this community. Center of Precision Agriculture of Arid University, they were also involved because they have the drones, and we use those drones. and the proposal was discussed and with UNICEF country office Pakistan for financial assistance. Series of meetings were held between the all three parties for finalization of the proposal, and finally proposal was approved by the UNICEF in July 2024, and we implemented in September 2024. September is a peak transmission period of the dengue. We have the first round of application of insecticide in September, then October, and the final is there just last week in November. So we have 3 application rounds. And for this identified area, we identified 8 large garbage sites were selected for the implementation of this pilot project, and 10 to 12 stagnant water bodies of the varying size were selected for the application of insecticide using the drone. Pre-application survey were conducted in July and August. 2 brands of the biocides, Bacillus thuringiensis Vectobac, which is actually granulars, and the Variant Biosciences, and we have the one local BT strains, and this is Pakistan's first indigenous BTI, which have been identified from local, and we also use this BTI just to make the comparison to what is comparison of the Vectobac and also local BTI strains to have their evaluation. Densities were monitored on the day 2, day 3, day 4, and then later weekly, bi-weekly, and 4th week each month. So we have the very good mechanism monitoring after the application. So this is a little bit a busy slide, but just gives you some results of the Vectobac. We have the 4 sites where we use this Vectobac, and we have the other 4 sites where we use our local BTI strains. If you look at this column, which give actually the pre-treatment survey, the positivity, very high positivity in this garbage. In the site 1, where the 28% of bottles were positive with Aedes mosquitoes, and site 2 had the 33.3%, 30%, which indicate that's very high positivity rate in this garbage site. After the application of this Vectobac, on day 2, we have the first survey that we make the 20 samples, and out of the 20 samples during day 2, the 4 were positive, which are the percentage 1%. Though is a very big reduction in the so yeah. Oh, sorry, it was yeah. So out of 22 samples were positive on day 2, and then to another 25 the day 4 there was 0 positivity, and the week 2 1 positive. And then this shows that after the application of this Vectobac, Bacillus thuringiensis, there was significant decline in the vector densities. And the same is with the site 2, with a 33% positivity. After the application, you can see the decline in the significant decline of the and then site 3, 30% before the application, and then you can see the 15% on the day 2, which is actually these 15% actually the first stage of first instar larvae, which immediately emerged from the and it was expected it will also be killed next couple of days, and which is true. And you can see that there's a significant decline. So all these 4 sites before the treatment shows the highest positivity of the Aedes mosquitoes before the treatment of the Vectobac Bacillus thuringiensis. However, after the application, there is significant decline in all 4 sites. Now we move to our local BTI. Local BTI, which is actually under the evaluation of the PQ team, WHO pre-qualification team, but it also shows a significant decline in the vector population after the application. In these sites, site 5, 6, 7, 8, 30% positive, 40% positivity in the site 6, and 24%. And after the application, you can see the significant decline of the Aedes larvae. So which means that this local strain of the BTA also showed a very promising results during this pilot project. Now move to this is actually not garbage site. These are actually our open stagnant waters, and you can see that we have the four we have the fourth site for this stagnant water. Previously, we have the garbage site where the heaps of the garbage, and now these are the open stagnant ponds. We have the 4 ponds in where we use this vector bag. And before the treatment, we have the 5 sample out of 5, 1 were positivity. So again, before the treatment, there was a very high positivity rate. And then again, after the application of this pond, there was a significant decline in the vector densities. And the same is case in case of the local BTI strain. In this site 6, where the pond which shows the high positivity rate in these 4 sites. But after the application, you can see that during the day 2, there is a zero positivity there, and then there was almost zero positivity after the application of the local strain as well, similar to the WHO pre-qualified vector bag product, which means that the both product BTI strains, local as well as the WHO pre-qualified, and this local BTI strain is also under the final stage under the evaluation of the pre-qualification team. So these 4 slides shows that the after the application of the BTI through using the drones, because if you look at I just go back to the one picture, this one, like this. How can you go upstairs? Because it's almost 2-story high heaps of the garbage. You can't climb go there for the treatment of our application of insecticide, or either is a BTI or chemical. So it is next to impossible to go there for the treatment of the. So there was only one option: the use of the drones for the application of the insect surveillance as well as the application of the insect, and up to this 2-story or the heaps of the garbage. And the same is here. And now you can see that the drone is the only option for in Pakistan scenario, particularly in this where it is next to impossible to treat these big heaps of the garbage for the application of any insecticide. So summary. Use of drone in the vector surveillance and control is an innovative and promising technology for overall management of vector-borne disease, particularly in the scenario of the Pakistan, where we are facing a huge problem of dengue regular outbreak just because of the inaccessible areas for the treatment of these solid waste. Both vector bag of the Valent Biosciences and BTI locals showed a significant decline in the larval population from day 4 to onward. On day 2, only the freshly hatched first instar larvae were collected while on day 4 and onward, only a few 1 to 2% sample were positive with the first instar. Actually, these why I put the first instar, because these are freshly emerged or have emerged larvae from the eggs, and which will expected to be killed in coming days under the influence of the BTI, which we applied in these sites. The control of Aedes mosquito is more significant in stagnant water compared to the heaps of the garbage. This is very important. People normally ask the question, where is the better result? And the stagnant water, where we applied the both types of the BTI, either local or as the vector bag, shows a significant in the stagnant water, because in the heap of the garbage, it was in some containers, it was difficult that the BTI reach in that container, and that's why there were very few positivity in these garbage heaps. And secondly, because there are very like a mountains where it is not possible to reach each and every bottle when there are hundreds of thousands empty bottles are lying there. That's why there are some very few positive, but despite this very few positive 1 to 2%, still we received a significant results in the control of the dengue vector breeding in this heap of the garbage. Drone can be used ideal for UNICEF, because we are also giving there is another window of thought that the UNICEF and some other partners who are working in the relief activities during the natural calamities like the disaster, like just like a flood, where the area is not accessible, and we can't deliver our commodities over there. So drone can be the best option to deliver those emergency kits, including the bed nets, into those inaccessible areas. Apart from the control of the dengue mosquitoes, we as a program giving a new window of thought to our partners that they can also be used this drone technology for delivery of their emergency kit in inaccessible areas during emergency like a flood which we have faced in 2022. This also includes the delivery of the IT and mosquito net to inaccessible areas. Again, we are very thankful to the UNICEF for giving this wonderful support for joining the hand to the national program for this first pilot project of Pakistan, which prove some very significant results for the country. And hopefully in the coming years, we will have some decline in the dengue incidents in country, particularly in the capital, where there are the big issue of the solid waste management. Challenges, despite our very good success in these, there are certain challenges like the regulatory and legal issues, because the use of drone often subject to the strict regulations and security clearance, especially in the populated and sensitive areas like airport. This also includes the security issues of the sensitive areas, even in individuals. Community leadership limitations. Sometimes the community raise the issue of and concern on the privacy of the family while flying the drones in their near neighboring areas. Data privacy and security. So these are the few challenges which we also identified. But on the other hand, what the success we achieved through this pilot project is a far ahead from these challenges. That's why we are, as a national program, we are in the consultation with the local authorities to have a better understanding of the launch of this pilot project, scale up this pilot project in other part of the country if the budget are available. But again, we found a very good results of the use of drones in the country. Technical limitation. Drones definitely requires a highly trained personnel to operate and maintain the drones. Cost of the operation. Definitely the drone is expensive intervention, and drone technology needs some high initial cost and maintenance cost. This may include the life of the batteries and pay or low capacity of the. So these are some operational challenges which we faced in this pilot project. But again, I would like to say that this is a wonderful technology, and that we can use, in particular in the areas where it is not possible to access for the human to reach there for the treatment of the areas. So that's why drone is the best technology for these such areas. So acknowledgement, thank you very much once again, UNICEF, for this wonderful support to the program, that APMEN, all the technical assistance, then Edge University, Chemtech, and some other partners who played a wonderful role for the success of this pilot project. And we are analyzing the data more critically. Definitely, you will ask some questions because the data is almost very preliminary, but we are compiling this data and analyzing and more statistically to generate some good evidence for the use of for scale up this pilot project in other part of the country, not this other part, but other areas as other countries as well. So thank you very much. I think I have done my job. Thank you very much.
Yes, thank you, Dr. Mukhtar. You made a very convincing case there about the advantages of drones to deliver products and access difficult to reach situations. So thank you for that. Very, very impressive mountains of breeding sites there. Steep rise in the number of dengue cases over the preceding decade. It's going to continue, absolutely no doubt about that, as a global trend. So I think, Luke, you might find a very welcome hand if you were to approach the Pakistan malaria control or dengue or vector-borne disease control people, in specific Dr. Mukhtar, if you're looking for sites to implement gene drive releases and trials and so on. So thank you, Dr. Mukhtar. We move on to our last presentation, which is by Professor Hung Wook Kwon, who will be talking to us about vector monitoring using imaging technology. Professor Kwon is director of the Center for Infectious Diseases and Vectors at the Incheon National University of South Korea. Among his many other positions and roles, he's also CEO of the technology company InSensory, which developed this AI-based insect species identification system for monitoring Anopheles, Culex, and Aedes, which he is going to be telling us a bit more about. Professor Kwon did his PhD in the Department of Neuroscience and Entomology, University of Arizona, followed by a postdoc at Vanderbilt University in Nashville. He now works on aspects of molecular and neural olfactory modulation in insect antennae and analysis of olfactory function in blood-feeding mosquitoes, among other topics. So, Professor Kwon, thank you for making yourself available. We look forward to your presentation. Over to you. Thank you.
Thank you so much. Let me share. Okay, thank you very much for your introduction, Dr. Brock. This is my great pleasure to talk about our recent study in technology advances in vector control. Even though this is a short presentation, if you have any questions, let me know. So I'm actually working at the International University right now. And as Dr. Brock said, we have a center as well as a startup company recently. So as you know, the reason why we actually study the vector-borne disease research is that there's a lot of changes in global climate change. So there's so many in Korea here also. We last summer we have a very hot summer and we have many mosquito problems and other problems in humans as well as animals too, the livestock. So as you can see, the climate changes keep raising now. And also there is some kind of dengue virus also. There's an in danger in Korea also, even though we don't have dengue virus here, cases here, but we still have many patients coming in from the outside every year. So that's why this is mostly, there's many disease vectors available around the world. Here we are focusing on malaria and dengue and many diseases and so many vectors. And the reason why is that there is, as I said, as you know also, the disease vector cases keep growing now. So we don't know. There's a lot of many factors actually affect on this number of cases now, but we should consider everything, but how we can do it. That's actually one of the questions now. So how can we prepare for the outbreak for the disease vectors and how we can mitigate the public health issues? So as you know, mostly there's mosquito control measures. There are so many classical ways and mosquito net, classical method and repellent and also insecticide and also mosquito killer, the lamp or something. But the important thing is that there are so many mosquitoes around the world. So this is actually in Siberia also, there are so many mosquitoes actually now present in the summer. So monitoring is not enough because so many mosquitoes are present here now. So, and as Dr. Brock said, I have been working on the insect olfaction usually with the mosquitoes and the Drosophila model, how these insects attracted to the host. So there are so many, the volatiles, CO2 and the sweat compounds, and also isovaleric acid. With this chemicals, mosquito actually use this chemical to attract to find the host, as well as the human the body temperature. So currently, to monitor the mosquito here in Korea, also throughout the world, we actually use some kind of black light lamp, as well as a busy trap to monitor the mosquito. And every year we go out to the field using these traps. We actually keep monitoring the mosquito population in Korea also. As you can see, this is the real case, right? So there are so many mosquitoes in some certain place. This is very hard to identify and isolate the mosquito species as well as virus or pathogens. It takes a lot of time. So that's actually a problem. That's why we actually try to develop an on-site real-time surveillance system, right? So this is the key for the future mosquito control measures. So recently, this is actually in Korea case, we actually have a digital mosquito monitoring system available right now since 2018. So this is the major cities, we have this kind of machines. Actually, this is not portable. This is very big and installed in certain place. So keep allowing to monitor the mosquito population in certain area. So you cannot move. So one of the problem is that if the environment has changed around this area and the mosquito population is changed. So that's one of the problem. And also, this is that currently this is not, this is actually using infrared. So to discriminate whether or not this is mosquito or non-mosquito species. So there's some kind of limit. And also this one from Netherlands and European countries, and using wind beat frequencies, and also the busy counter. This actually machine use wing beat frequency of mosquito species to discriminate the mosquito species. And this is another thing. And also some other machines also, they use other mosquito, as I said, wing beat frequency also. But if you can actually see the number of mosquito collected in this machine, is you can actually see the male and female, there's a difference between sexes. So maybe actually different from this kind of, there's some kind of limitation currently. So we have to keep developing this kind of machine now. So, and also, as I said, there's other technology using some kind of infrared rays to discriminate the wing beat frequency. When they pass this beam, these machines actually discriminate the mosquito species based on wing beat frequency. So even though you can actually see here, there's an increase of accuracy in comparison with the classical way, we still need upgrade the quality of these kind of machines to monitor the mosquito population better and efficiently. So what's the solution for the next step? So as the first speaker said, the deep learning technique now is very prevalent. And we should try to incorporate this technology to monitor the mosquito population. So as Dr. Brock introduced me, I studied the neuroscience using mosquito and the insect model. So this is typically what happened in the neurons of the the animals, the human, and also mosquitoes, also same. You can actually see, this is one neuron, for example. There's a lot of inputs coming into the cell body. And the stimuli, these stimuli actually sum in cell body. And then this cell actually decide whether or not they actually elicit some signals to the axon. So this is actually exactly the same in deep learning technology. You can actually see there's a fine sum here. This is like a cell body in the neuron. there's a many informations coming in, and then this the calculator decide whether or not they actually have some kind of activation signals to the next. This is exactly what the artificial networks actually explain. Now, this explanation was invented actually in 1958. So that's why several years ago, our lab has been working on collecting some mosquito images in the lab. You can many maybe 6,000 images in each samples, and this is enough to make some kind of model. So Aedes albopictus, Aedes vexans, and the Anopheles species complex, and then Culex pipiens and Culex tree, which is actually major vectors in South Korea now. So this is the model in the DCN model. You can actually see we have this kind of photos, and let the machines actually learn this kind of pictures. This is actually published in 2020 in Scientific Reports. Interestingly, this machine, this deconvolution neural network, this algorithm, also they are focusing on the some kind of special features, morphological features from the mosquito. So this is actually visualization where these machines actually focus on specific morphology. So this is, for example, this is Aedes albopictus. They focusing on some kind of stripe on the thorax and the abdomen. So you can, the red color means this machine really looks at the specific morphological features. So based on this, you can actually see by the training, this DCNN models, they now learn this is Aedes albopictus with the 96.5% accuracy. So this is also same. They looking at the stripes on the abdomen and thorax area. And this is Anopheles species. You can actually see there's morphological features on the wing. These machines take especially focus on the wing patterns. You can actually see this. So, okay, and then based on this, we are actually trying to develop two machines now. One is mosquito captures, and the other one is mosquito sorters. So with this one, we try to, this machine knows using this kind of pictures, they have some kind of very, very accurate score to identify the mosquito species. So typically there are three algorithms. So we use the three algorithms, and you can actually see ResNet is they show the best score. Maybe actually very similar, over 95% of accuracy, but you can actually see using ResNet 50, this is very, very accurate. So based on this, we now still collecting many images to increase the quality of this technology, the machines now. And this is what we invented recently, and this is most captured. This is very small size, and compared to previous one, this is very small, and also this is very. You can actually take this to the field. This is a portable, and the mosquito coming in through this area, and then this machine take a picture with a web camera, and you can actually have this kind of quality of the mosquito captures. So this machine with the learned algorithm shows that there's this is Culex pipiens 90.97% accuracy. This is Anopheles species 99% or 90. This is not mosquito, so this is others. So they can also discriminate between mosquitoes and non-mosquito species. And there's one of the problems still. So we still have many non-mosquitoes collected. So we have to add some specific attractant and something like that to increase these numbers. So, but basically, we can collect the mosquito species here, and then this machine identify the mosquito species in on site in real time. So using this kind of monitoring system, we actually have now, this is the classical way of pest control. Using this data, real-time data, we can use eco-friendly way of pest control measures. So we can protect our environment as well as decrease the mosquito population with a very reasonable way. So also we can decrease the time to identify the mosquito species also using this one. So using this kind of technology, we can decrease the cost reduction in healthcare as well as improve the quality of life as well as cost saving, because we can decrease the amount of insecticide and also the human time input. And also we can apply these machines to the livestock. So next year we try to expand this service to the livestock to decrease the dangerousness in the livestock. This year in Korea we have many lumpy skin disease problem in cow. So we can. It turns out that mosquito is a major vector in lumpy skin in cow. So we try to apply this kind of things to here. So also, okay. And using this kind of machine, we have, as I said, we have a mouse capture. This is real-time mosquito capture machines. And also we have a mouse sorter. We collect many dead mosquito outside and we bring this to the lab. And then we spend many times under the microscope to identify the mosquito species. But using this machine, you can just go home. And then next day, maybe the idea is that this machine identify and allocate the mosquito species automatically. This is the idea. And then we try to develop some vector site with which we can apply to the pest control measures. So this is the idea. So I think that in IPM to mosquito control, we now actually this presentation, the gene drive using the genomics and the big data, and also AI application and eco-friendly way, as well as attractant and the specific attractant and repellent. We can develop now the advanced integrated pest control, pest management in mosquito control. So this is the new area to convergence research in mosquito control. So this is actually a long way to go, I think. But with this kind of tight collaboration, we can actually make a new research area as well as development and industrial development as well. So there's, as I said, there's many challenges and consideration on AIPM with the big data. Is the data quality is very important, because in the lab, the 97% accuracy, for example, in the lab is not always true in outside in the field. Field usually 80% to 70%. So we have to increase the data quality, and also there's a privacy security issues also. And also technical infrastructure requirement, because so many technologies actually fused together. So we still aware that there's many limitations. So we have to overcome these kind of things. Okay, so in summary, the big data and the recent the advanced technology actually enhance the environmental preservation effort. And also the using from the climate change, the big data actually provide the insight to protect and sustain our environment efficiently. And also using this kind of technology, we need a strong collaboration regionally. For example, AIs, actually we train the mosquito species in each countries. So we need a strong collaboration from the region. Okay, so this is our lab, and we currently have many supports. And then, okay, thank you very much.
Well, thank you so much, Professor Kwon. I think everybody in the audience is well aware that you need effective vector surveillance to be able to implement effective vector control. Effective vector control is absolutely based on effective vector surveillance. So, you know, the system or approach you explained here, you know, holds great promise to automate, speed up vector surveillance, and also avoid some of the problems, you know, where the gold standard has been somebody sitting on a chair for multiple hours and with an aspirator collecting whatever mosquitoes are biting you. And, you know, there's always the risk of not just malaria, but also arbovirus infection and so on. So this would bridge that kind of problem. So very interesting technology. The question that comes up in my own mind is it would be very good to have some experimental evidence, published evidence that demonstrates the actual comparing the current situation as mostly practiced by vector-borne disease control agencies as compared with your techniques. How closely do the results correlate with each other? So, but certainly very promising technology. Thank you, everyone, for those wonderful presentations. I've been going through the questions and I'm just nitpicking going along the way, and I'm going to try and spread some of the questions among our panelists, starting with Peter. There's a question here. Hi, Professor Hadawi. I'm currently working on some dengue risk mapping in Cambodia. How recent would the Google imagery need to be useful? I think you explained that in your presentation, that Google Earth and so on, it's a year, 2 years lagging behind current date. So that has advantages, but obviously it also has disadvantages. And then the second part of his question was, have any other countries beyond Thailand tried using this specific approach that you demonstrated to us? So the first one is related to, is there a way where you can get more current imagery than Google Earth that tends to be a couple of years out of date? And the second one, how widespread is your technology applied?
Yeah, okay. So, yeah, so in terms of the date, I mean, it varies a lot. You know, you can get, because Google is just continuously updating Street View images. And so you may find an area where the images are 3 years out of date. You may find an area where the images are from the current year, right? There's no sort of regular update schedule where you can anticipate it. But I mean, we have done studies where we've used Street View images that were not from the same year as our, say, larva counts. And it worked fine. So, you know, basically the idea is this. If you're talking about where is there a specific container, so is a tire still in front of this particular house? Sure, that's going to change pretty fast. But if you're talking about the number of containers in a district, right? If your area is large enough, that's not going to change that much from year to year. Now we don't have we are actually now conducting a study of this to look at what is the rate of change of container counts over time, because we gathered some Street View images a couple of years ago and we're gathering them now, and then we can do a comparison at least in one area. But that's the idea, that if the area is reasonably large, the container counts are not going to change too much. The other question, sorry, I forgot the other question.
How widely is this approach
Oh,
applied beyond Thailand?
right.
Do you have any
Yeah,
idea?
so one thing is, when we use this approach, we need to know what we're looking for. And so as you observed, you know, we're focusing on the most productive containers. So we basically go to the literature and go to the medical entomologists and ask them, in this region, what are the most important containers? And then we build the model to detect those. We have done this in, you know, several provinces in Thailand, and we've also done it recently in Rio de Janeiro in Brazil, because we were contacted by a group actually at University of Heidelberg that had a project in Brazil, and they wanted to use our technique to combine it. They were using satellite data, and they wanted to combine this with the satellite data to get container counts and then compare them to larva counts. So we have done this in Rio. And again, the types of containers vary from area to area. So in Rio, it was storm drains and large trash bins, and a particular kind of trash bin, smaller one there, that you know, that you don't have in Thailand. In Thailand, you get these big ceramic jars. You don't get those in Brazil. So it varies from area to area, but the technique works fine. The only thing, you know, we can't detect containers that are very, very small. They're just too small to detect. But that seems to be all right. If you wanted to do something like detect the landfills that we saw in or these garbage piles that we saw in one of the talks, then it's not object detection, it's scene detection, it's scene classification, which one can also do, right? It's a slightly different technique, but it's still using a convolutional neural net.
Yeah, you know, I just think that your approach also offers opportunities for why are there certain trends? So Aedes aegypti is more an urban vector than Aedes albopictus, which is typically more peri-urban rural.
Yeah.
And dengue incidence is higher in urban environments associated with higher levels of aegypti. So why is it that aegypti prefers a rural environment and albopictus a rural environment? What causes the switch? And is it breeding site related, which is where your technology comes in? I'm hoping you will link up with Bichara, because it is a question that we're interested in.
We do a lot of work together.
Oh, that's great.
Yeah.
That's great. So let me just go. Thank you for that, Peter. Christopher, here's a question for you. Is this method feasible and realistic to put into practice in resource poor areas? I just, you know, basically, fundamentally, the question is, how specialized and how expensive is the equipment that you require to apply this kind of approach that you're studying? So can you give us a sense of infrastructure and equipment cost, or what do you need to do what you do?
So currently, for the application and to use the application, we put in special effort to design the application from the start to be something which can be used with a very low threshold and which doesn't like require specialized equipment for it, because as you already see, like we can build like really nice imaging towers, and I think those are like really great solutions. But I think we should go maybe also through different approaches, and our was to just have a solution which would be as cheap as possible. And for currently, so if you want to use our application, you would only need a computer and smartphone and a macro lens, which you can get for around $5 to $10, and some pair of tweezers to remove the wings of the mosquito. And then you can like just image I think I've like shown a small image in the presentation. You can just put on the wing on the Petri dish and then put your phone on it and image it. And either you could install the application on your own computer and let it run there, or exit it through our web server and then upload your images, and you would get a response to it. So this was the idea, because we are aiming to especially use this application in a low resource setting, because in settings where we have maybe more resources, then we could go for such imaging towers, as we also seen, which I think really nice. Yeah, but generally, to be like cautious with using this application, of course, is that the models are only able to classify and identify the species which we've shown images to them. So you can't just go out and just apply the model everywhere, and it would work everywhere. But you maybe have first to get an idea of the species distribution in your area and understand, okay, is the model able to, there's a model already trained to grasp this distribution and to classify this distribution, or are maybe important species missing? Because it's really hard for the model to recognize, even if we train it on it, to recognize a species which has never been trained on. And therefore, if you go to a completely new setting, then you first maybe have to do some small feasibility study, or maybe like a small study to see, okay, what is my distribution, and then see if the application is working for you. But we are still working on to improve this and to improve that you can more easily use this application into new contexts.
Yeah, thank you for that, Christopher. That's helpful. So, Luke, I'd like to ask a question. There's a question here about whether there's merit in doing a stepwise approach, first population suppression, and then bringing in population replacement. That's probably not a very practical, cost-effective approach to take. But I'd like to ask you this question, please, Luke. So, how, there's a need to combat malaria vectors, and there's a need to combat arbovirus vectors, in specifically Aedes-borne viruses. So you seem to be focusing on Aedes. So let me ask you this question. How close are you, how ready are you to actually deploy this gene drive technology for Aedes aegypti suppression or replacement? And from the moment you find a partner and there's funding available, how ready are you? But also, how long will it take for you to actually release the first mosquito? And what is the reason for the gap between commitment in terms of country willingness and funding availability? What are the reasons for the delay? Is it because you need to establish infrastructure? Is it because you need to go through the whole legal process, regulatory process in the country? Is it because of trying to get public consent to actually use genetically modified organisms in that country that people are very worried about unwanted forever after consequences? Or is it all of the above? Your responses, if you can, Luke. Thank you.
Great questions. So in terms of the timescale for technical availability, we have this sort of super SIT enhanced sterile mail system, I would say, ready for field action, which would obviously be trials first. Now, gene drives are for Aedes are a little further away. There's a key technical limitation about homing rates being lower in Aegypti than Anopheles for the aficionados. Time, it's absolutely overwhelmingly regulatory political community issues. So producing the eggs is quick and cheap, and if there were no regulatory political community issues, you could go out and do it. I won't say tomorrow, but really very quickly. Clearly, for any new intervention, you will need community support. People will want it, and there will be some sort of regulatory process. And there is for drones, as was mentioned in the previous talk, as there would be for a new insecticide or a new vaccine or whatever. And there certainly is for large-scale release of genetically modified insects. But it doesn't necessarily, so it takes some time, but not necessarily as much time as you might think. So just to go back to my Oxtec days, we made a transgenic mosquito, our first transgenic Aedes aegypti in 2002, and we had the first field trials in 2009, and that was the first release of a GM mosquito anywhere in the world. So you can imagine that the sort of regulatory and community and all these issues loomed pretty large in that, and yet it was still not that many years from making the strain. And that's before actually testing it in the lab and persuading ourselves that it was a useful thing to go into the field, and then actually getting it out into the field. So it doesn't necessarily take as long as some people think, but it is definitely an issue. It's the regulatory, legal, political, community side, and you know, appropriately, I think. But people want to be cautious, and all these things have to be looked at very carefully by independent third parties.
How much, what kind of cost are we looking at, Luke? You know, is it Wolbachia kind of scale, or you know, dollar-wise, is it a
yeah, but I mean, different approaches have different finances. So basically, the more invasive the gene drive, the lower release numbers, and so on the whole, you will have higher technical development, regulatory, political costs to persuade people to let you do it. But then actually the deployment in principle is releasing really very small numbers of mosquitoes relative to the target population.
Yeah.
More controllable, reversible methods are going to be intermediate. So by their nature, you will inevitably need to release more, but not as many as you do for current sterile male methods. And I mean, really, this is part of your sterile male methods work really well, but you need to release very large numbers very, very frequently. And so the motivation for many of these other methods is to try to get much of the same clean, species-specific, targeted impact, but with lower release numbers. And that's our enhanced SIT and various forms of gene drive as well.
Yeah, thank you for that, Luke. I mean, we could carry on talking for hours. It's a really fascinating field. And I thank you for the time that you've spent with us. Dr. Mukhtar, there was a comment here by Prudence, who said, just as a matter of interest, in some countries in Africa, they now starting to invest at larger scale in rice production. And obviously rice fields, especially wet rice, leads to massive increases in mosquito breeding, including Anopheles, malaria vectors. So, you know, and in a place like Rwanda, they're now also investing in drone technology to combat the increased expanses of malaria vector breeding. There was a question from the audience to ask, how do you actually deliver the BTI, the insecticide or product that you want to deliver? Is it kind of a remote? It's got claspers and it holds the bag of whatever. And then you have a remote button, and when you hover over a site, you just press the button and boom, there you go. How does it work? Just briefly, Dr. Mukhtar.
Yeah, thank you very much. It's a great question, and many people ask this question about the delivery of the insecticide. Yes, actually, we use the water formula, granules, which we dilute in water, and there are specific containers which are fitted with the drones, and those containers contain this water formulation, and there is very controlled release of the insect this liquid through a very specific nozzles. So it is actually the water soluble of the granules which we dilute in water, and there are specially fitted the bottles containers with the drones, which and those bottles have a very controlled release of insecticide according to this area. So it's not a powder form or the other formulation which has dusted through the drones. It is actually water based solution which we diluted in a water and applied through the drone. I think this is the question.
Correct. Yeah. So, Professor Kwon, I'm very sorry. I feel extremely guilty that I'm going to neglect you a little bit, but we have very definitely run beyond our allotted time, and we have to bring this webinar to an end. So I'm going to ask Dr. Pon or Popo, please to put the poll questions up on the screen. Each individual, when they tick the various boxes, it will go against your name. So you don't have to worry that everybody is going to use the same form. It's individualized based on your particular link. So I'd be very grateful if you can just respond to those few questions. And while that is going on, please let me make use of the opportunity to extend a genuine, deep word of thanks to our various panelists. You've been extremely patient. I know you are all very busy people, and you've made a significant sacrifice by setting aside these 2.5 hours in preparation and actually taking part in the webinar. So thank you sincerely from the APMEN team, from our audience members. I'm sure our audience members learned a great deal, as have I, and enjoyed the interaction and participation. So thank you to you, also to Dr. Pon and Popo for your backroom help with the technical side. And finally, to our audience members, wonderful to have you participate in such a highly informative webinar. Thank you for your patience. Thank you for linking in. And then we will have more webinars starting again next year. And we look forward to, again, engaging with our vector control community for additional webinars next year. To all of you, sincere thank you, and go well. Thank you.
Thank you very much.
Thank you. Thank you, Leo, for organizing this really wonderful event.
Thanks so much.
Thank you all for wonderful meeting.
Thank you,
Thank you
Professor
very much,
Kwon. I
everyone.
apologize. Yeah.
Thank you very much, Dr. Leo, Papa, Dr. Fon, for this wonderful webinar.
Thank you,
Thank you
Dr.
very much.